ورحمة الله وبركاته. أحب أقدم نفسي الطالبة روناك قيصر من جامعة الكرخ العلوم قسم أدلة جنائية مرحلة ثالثة. طبعا عنوان البرزنتيشن مالتي يكون DNA and DNA extraction. المحاضرة حتكون كاملة باللغة الإنجليزية. I hope you enjoy it. So first, what is the DNA? DNA is deoxyribonucleic acid. It is the molecule that is contain the biological instruction that is make each species unique. A DNA along with this instruction is contained and is passed from adult organism to their offsprings during their reproduction. And it is composed of a nucleotide, which are the basic building block of the DNA. And each nucleotide consists of sugar molecules, phosphate group, and one of the 4 nitrogen bases, adenine, thymine, guanine, and cytosine. And here we have the structure of the DNA in double helix. And here we have the nitrogen bases. The nitrogen bases bound in hydrogen bound, and sugar molecules is bound with the phosphodiester bound. And a nucleotide consists of phosphate group, sugar, and nitrogen base. I mentioned that. So where we can find the DNA? We can find the DNA inside we can find DNA in almost all living organisms from bacteria and viruses to plants and animals, and in a human. DNA found in every cell of the body, including the skin cell, blood cell, and organs such as the liver and brain. Here we have a figure that shows us where we can find the DNA exactly inside the cell, inside the nucleus in the cell. So, and here we have video that show us where we can find the DNA exactly. Here we have a cell inside a human body, and it contains cellular membrane and cytoplasm. And mitochondria, it's called the house of energy. And follicles and Golgi apparatus. And here we have the nucleus, which is contain the DNA, and DNA surrounding or wrapped around the protein histones, which is form the nucleosome. And within the DNA, there is a section called the gene. Gene, it is the basic unit heredity in the DNA. And DNA, it is the deoxyribonucleic acid because it is linked to oxygen atom, and it is a double helix in structure. Here we have the sugar phosphate background, and we have a nitrogen bases. This is structure of the adenine. This is structure of thymine, and this is structure of guanine. And here is structure of cytosine. And these, what are the consists of a nucleotide? They form together and form the DNA in form of double helix, like this video. So after that, what is the function of the DNA? The function of the DNA, it is to store and transmit the genetic information from one generation to the next, providing the blueprint from the development and maintenance of the living organism. And one of the primary function of the DNA is to provide the code for a synthesis of a protein, and protein are essential molecules to carry out many of the function of the living cell, and including the catalyze the chemical reaction, providing the structure and support, and facilitate the cell communication, and the genetic code carried by the DNA is read by a cellular machinery, and that's used to synthesize the protein by linking together an amino acid in a specific order. That is sequences of this amino acid determine the structure and function of the protein. After that, we have the purpose of the DNA extraction. Why we extraction the DNA? The purpose of the DNA extraction is to isolate it and purify the DNA from the sample of interest. And once the DNA has been extracted, it can be used in a variety of downstream application, and it is include the PCR amplification, DNA extracted from the sample that can be used in template for PCR amplification, which is allowed for a specific amplification of the target sequence's DNA. And we have, and the second one is the DNA sequences that can be used for sequences determine the sequences of the DNA. And the genotyping, the DNA that is structured from the sample can be used for genotyping, which is involved the analyzing genetic variation in individual DNA. And the gene expression analysis also, the DNA that is extracted from the sample can be used to study the gene expression pattern in different tissue or under the different condition. And the last one is cloning. The DNA extracted from sample also can be used in cloned in a vector from further manipulation or analysis. And after that, we have the DNA isolation. The DNA isolation, it is the process of a purification of the DNA from the sample using the combination of physical or chemical method. The DNA can be isolated from the nucleus. And there is some problem that is when we want to extract the DNA or isolate the DNA. First, the DNA is found in a nucleus, which is surrounded by two membranes. And two, that's the presence of the DNA, which is degraded the DNA, and DNAs, which is degraded the DNA, and the presence of the micromolecules. And there is 4 steps to remove and purify the DNA from the rest of the cell. First, we have lysis and precipitation and washing and resuspension. So here we have the method of the DNA extraction. First, we have a magnetic bead method. This method that involves the use of a magnetic bead coated with the substance that selectively bound to the DNA, and the DNA bound bead are then separated from the other cellular compound using the magnetic, and the DNA is evaluated from the bead. And here we have the figures show us how this method can be done. First, we added the magnetic bead, and then it will specific banding in magnet, and DNA will bond. And then we remove the supernated, and we wash it with the, first we precipitated with the ethanol, and we washed it with 7% with ethanol, and we transfer it to the clean tube, and we do polymerization reaction or a flip to amplification the DNA. And the next method is the organic extraction. It's called the phenol chromoform. This method involves the use of phenol chromoform to separate the DNA from the other cellular compound. The phenol is used to lysis the cell, while the chloroform is used to remove the protein and lipid. And the DNA is precipitated and washed with the ethanol and remove any residuous contamination. And here we have this figure show us how this method can be done. First, we lysis the cell by adding a lysis buffer, SDS and proteinase K. SDS is used to solubilize the lipid membrane and denaturate the protein, and proteinase K used to denaturate the protein. And then we incubated it, and after that we added the phenol chromoform, and we do vortex and centrifuge that will separate the sample in 3 layers. The first layer is the DNA, aqueous layers contain the DNA, and the second layer is protein, and the third layer is lipid. And after that, we transfer it to the aqueous layer to the clean tube, and we precipitate it with the ethanol, and we wash it with the 70% also ethanol, and we remove the supernated, and we transfer it to the clean tube, and also we do PCR or a flip to amplification the DNA. And the next method is the Chilix. Chilix method, it is the resin composed of styrene divalent benzene copolymer that is containing the per-amino diacetyl ion that is act in a catalytic group in binding polyvalent metal ions such as magnesium. And the basic Chilix procedure is pulling the sample in 5%, and the Chilix solution that will lead to disrupting the cell membrane as well as destroying the cell protein. Here, the figure shows us the Chilix method. First, we added the water, and we added the Chilix resin. And then after that, we pulling the sample and centrifuge, and we transfer it to the cleaner tube. and we precipitate it with the ethanol, and we're washing it also with a 70% ethanol, and we transfer it also to a cleaner tube, and then we do PCR and or a reflip to amplification of the DNA, and of course the quantity and purity of the DNA check. And after that, we have the FDA paper. It is another method that's used in forensic lab to extracting the DNA. It is composed of a cellulose paper that is contain the chemical to protect the DNA from degradation by the nucleus. And amplifies on the FDA paper can be stored for several years. What's happened here is we place the blood sample and or apply it on FDA paper, and cell will be lysed immediately by the chemical that found on FDA paper. And piece of this paper is taken to wash and purified by a solvent and remove the heme from blood because it interferes with the PCR and inhabitation PCR. And this method is excellent because it does not require the DNA amplification. This figure shows us the FDA paper, how it look like. So after that, we have to know what is the PCR. PCR is polymerase chain reaction that is used to amplification the DNA. We have the steps, basic steps for PCR. Denaturation, it is the first step is to denaturate the double-stranded of the DNA template by heating. So at high heat temperature, typically to 95 for a few seconds, and that will break the hydrogen bond between the complementary strand and separated them into the single strand. And the next step is annealing. At the temperature, it is then allowed typically to 50 to 60 Celsius, and allowed to primer to anneal to the complementary sequences on each strand of the DNA. The primer are short DNA sequences, usually between the 18 to 25 nucleotide long, and that are designed to match the target sequences to the region of interest. And the last step is extension. Once the primer are annealed, the temperature is raised typically to 72 Celsius. The DNA polymerase enzyme is added to extend the primer and synthesizes the new DNA strand. And the DNA polymerase is used to a single strand DNA as a template and a complementary nucleotide to extend the primer. And after that, before that, and also after we have the repeat the cycle to denaturation and annealing, the extinction step and extinction step, and repeat this multiple times, usually between 20 to 40 cycle. And each cycle is doubling the amount of the DNA in reaction and exponentially amplification the result in the large amount of the target DNA sequences. And the final extension, it is after the final cycle, the long extinction steps typically between the 5 to 10 minutes at 72 Celsius is added to ensure that the remaining single stranded DNA is fully extended and ensure that all the DNA fragments are the same length. Here we have this figure show us how the PCR can work, and this is the step in PCR. First, we have the DNA in double helix that from 5' to 3' and 3' to 5' opposite direction. First, we have the denaturation. It is separated the double helix of the DNA to a single strand, and then the annealing, the primer will conjugate it to the target sequences of the DNA, and in extinction, the tag polymerase will extend the strand of the DNA. So we have a type of PCR. After we know what is the PCR, there is the type of PCR. First, we have a strand PCR. It is the most basic type of PCR as described in previous answer. So it is used to amplify the specific DNA sequences from the complex mixture. And the next one is reverse transcriptase PCR. This technique that is used to amplify the RNA instead of the DNA. And the RNA is the first convert to the complementary DNA and use in reverse transcriptase enzyme, which is then amplify using the PCR. And then we have the quantitative PCR. It is known also the real-time PCR, this technique that is used to quantify the amount of the DNA or RNA in the sample that is used in fluorescence dye or probes to measure that amount of amplified DNA or RNA during the amplification process. And the next one, we have a digital PCR. This technique is similar to quantitative PCR, but the partitionally the sample into the many small reaction to allow the detection of a rare target with the high precision. And we have the next one, nested PCR. It's used to a set of primer. This technique involved a round of PCR amplification with the second round that uses a set of a nested primer that is bound within the product of the first round. And the result is increase the specificity and sensitivity for the reaction and low abundance targets. And the multiplex PCR, the technique allow for the semi-continuously amplification of multiple targets in a single reaction use the multiple set of primer. And this type of PCR use depend on the specific amplification and the type of the DNA and RNA being amplified. So, and after that we have gel electrophoresis. Gel electrophoresis is this technique that is used to separate the DNA or protein molecules based on their size and charge. And basic steps for electrophoresis is preparation, the gel. The gel are made of polymer such as agarose or polyacromide. It's prepared in a model with the walls at the one end. And the gel is then submerged in the buffer solution that conducts the electricity. And loading the sample, the DNA or the protein sample that is mixed with the loading buffer that is contain tracking dye and then loaded into the walls of the gel using the micropipette. And the electrophoresis, it is the voltage that is applied across the gel using the electrode placed at each end of the gel. And the negative charge of the DNA or the protein molecules are attached to the positive charge of electrode and migrated through the gel toward the negative charge electrode. And the small molecules move through the gel more quickly than the large one. And resulting in that is separation based on the size. And the standing, it is when the electrophoresis is complete, the gel stand with the dyes such as the ethidium bromide to visualize the separation DNA or the protein that's bound under the UV. And the gel electrophoresis is widely used technique in molecular biology, in biochemistry, and for separation and analyzing the DNA or the protein molecules that's based on their size or charge and their charge. And this can be used for a variety of application, including the genotyping, gene expression analysis, and protein purification and the DNA sequences. And after that, we have this figure of gel electrophoresis. After we extracted the DNA and we take it to PCR to amplification the DNA, and we take it to gel electrophoresis because we can't see the DNA because it is transparent. So we put the dye ethidium bromide, and it shows us this photo. It is separated the DNA molecules based on their size and charge. And this is the end of my presentation. I hope you enjoy it. And if anyone have question, please ask.
رائعة روناك، رائعة. ما شاء الله عليك. أسلوب وإداء ولغة ومعلومات وطريقة عرض جدا رائعة. كل التوفيق.
شكرا لك دكتور.
الله يحفظك. يعني يمكن الأخوان كتبوا بالشات يمدحون بك قبل ما يخلص، ما قدروا يوقفون يعني. الله
شكر جزيلا لهم.
الله
تسلم
يحفظك.
دكتور.
تابعي الشات وراح تشوفينه. دكتورة هدى أو دكتور لؤي، تفضل.
السلام عليكم ورحمة الله.
عليكم السلام دكتور.
Actually, I have one question. What is the difference between a primer that used in DNA replication and used in PCR, polymerase chain reaction?
First, we use a different primer to any DNA extraction. Like this primer, like if you want to copy the DNA, we have to use a specific primer to that one. Like, for example, if we want to analyze to COVID-19, we have to use a primer for COVID-19. So that will give us the result incorrect.
No, actually, I am asking what is the difference between DNA used in PCR and used in DNA replication. The difference that in DNA replication, it is RNA. And
RNA.
excuse me, let me
finish. Yeah, go ahead.
And in PCR, it is DNA. Okay, thank you very much.
Yes, I mentioned that I told you that we have to use a specific primer to each DNA or RNA extraction. We can't use the same primer, so I think it's the difference.
Also بالعربي راح احكي لك انه بالدي ان اي ريبليكيشن البرايمر هو RNA، ما له علاقه بالسبيسيفيك او نون سبيسيفيك، والبي سي ار هو DNA. الفرق هذا DNA وهذا RNA.
نعم دكتور.
سبيسيفيك طبعا لكل جين.
يمكن فهمت سؤالك غلط.
نعم.
أقول يمكن فهمت سؤالك غلط وجاوبتك بإجابة غلط.
على كل حال مشكورة وعاشتي لك.
شكرا لك دكتور. شكرا لاستماعك وشكرا لحضوركم.
اوكي.
طبعا احب اشكر دكتورة
لحظة روناك. تفضل. نعم. دكتور.
شنو الفرق بين التي ام والتي اي؟
التي ام والتي اي in DNA extraction؟
عندك اي نعم.
Actually I have no idea about that.
انت تكلمت عنها بالبي سي ار، عندك الملتنج temperature والانيلينج temperature.
Yeah, that's the step of PCR. First, we have denaturation and annealing and extinction. In denaturation, it is the high temperature, 95 Celsius. So in this temperature, the DNA is separated from double helix into the single stranded.
Do you have an idea how to calculate the TM and TA?
It is the technique in PCR.
Random.
This is a technique in PCR, Professor.
We have to calculate it when we design a primer.
We control the cycle in PCR first.
No. When we design a primer, to be specific, we have to calculate the TM and we have to calculate TA. There are formula specific for these terms for the melting and annealing. Okay, thank you very much. Thanks.
شكرا.
شكرا جزيلا. شكرا جزيلا.
أحب أشكر الدكتورة أسماء ودكتور فراس ودكتور العميد الكلية مالتنا براق لأن تواجدهم اليوم ويانا واطلاعهم على إلقائنا للمحاضرات. أنا جدا أشكرهم لدعمهم، وكذلك دكتور أثير أنت حضرتك إلى كل يعني دور كلش كبير بدعمنا بإلقاء المحاضرات.
تستاهلون. نفتخر بكم. دكتور براق موجود؟ دكتور براق يا ريت إذا موجود دكتور براق